So that's all. That's the basic that you should know. And coming to the regulations. So generally, like when we own acronym, like for generally we never that humans, we say that as in a single term. So what they did is like in the year 1960, and it is they create as gathered there, and they had a meeting about this genetics, because like they have to make international standard for chromosome nomenclature. So what they did is like they in the year 1960 in the Denver conference, these scientists were sorting criteria human chromosomes, which comes first, which comes second, which comes third, because they in this conference only they decided like the biggest comes first and the metacentric will be first, metacentric second chromosome, second, third chromosome. So decided like the sorting area for, and terminology for that. So that it is in French, it is called as legume, which means small. P, it is like a Q. So just they randomly they made it as long arm. So after small, it is long. It's like an ascending way. So understand manually, and then you have to go with the software things, because software makes mistakes. So more enough, then we can able to identify which software able to predict it. So acrocentric, we say it as Y. Autosomes is body chromosomes, allosomes is a sex chromosomes. That is 22 pairs is called as we call it as autosomes. It is a body chromosomes, and allosome is sex chromosome, which we decide male and female. This is what the terminology says. Like, see the important slide that you should know, or else completely shocked. Like you seeing like 30 you not by explain and like you know the everything this mechanism chromosome present inside the sleep. You have to culture the blood. So what we'll be doing, like later I will say, like amniocentesis and other things and all, like it comes under like genetic diagnosis, I'll say in the, but generally what we will be doing, like you understand the basic concept. I'm not going in detail, just you understand the basic concept alone, because like what we will be doing, we will be isolating the chromosome from the blood, because like we will be taking blood from the body, and we will be culturing the blood in the let's say so how I can say is that, like because I don't. Okay, you know this meiosis. So meiosis by providing all the requirements. So what we'll be doing, like we'll be arresting the cells during the metaphase stage. To reach the metaphase stage, bit confused rather than seeing it. But I will say in this way so that you can able to understand. This is a cell culture thing. You can see, everyone know, like probably the post graduation students might have known what is mean by laminar or laminar airflow. This laminar airflow, like if you are not aware, I will say, like because to avoid the contamination for this microbiological process or for this culture, we use food to avoid the contamination. We have a separate lab to do this. Other than this culture, we never do anything. So this is how it works, and you could see the chemicals that is present, and we'll be adding 4.5 ml of RPMI and 1.5 ml of PHA and vitamins 100 ml and antibiotics 100 ml, 100 ml. Because like after that only the growth phase starts. So that here you could see, like G1S, G2, M, you could see anaphase, telophase, everything. Because you are familiar about this mitosis, I don't want to give glance what it means by mitosis, everything. So it takes more time. So you understand this phase, and what we have to do is that the procedure is to arrest the mitosis to attain the metaphase stage. So what we'll be doing at the 72nd hour, 3 days experiment, we'll be adding all these things, and we will be putting this in the CO2 incubator, which means it will be having 36.8 degrees Celsius according to our body temperature. This incubator will be set it up, and it is a fixed one. We never use for any other things to avoid the contamination. You can use this, and every 12 hours we will be taking out, and we will be giving a mixed. We will be giving a gentle shake, so that because like the blood and the other plasma will be settled down, and the chemicals will be in the top position. To avoid that and to give the aeration, we will be opening the cap little bit, and we will be shaking it up, and you could able to and so this is like rather what I'm saying is a little bit complicated. When you see by IC, it will be very clear to understand, but just you have the idea of that, so that you won't be whenever you're doing, whenever you're coming crossing this experiment, you won't have any confusions. So what we'll be doing is that, like, we'll at the end of the 72nd hour, that is the third day, we will be putting 100 ml of colchicine to arrest the metaphase stage, because in the metaphase stage very invisible. So we'll be adding colchicine, and we'll be incubating for 1 hour, and again we will be doing this harvesting process. We keep it in the centrifuge, we discard the supernatant, and in that part there will be a pellet, and we will be adding 6 ml of 0.075 mkcl, which should be preheated. So we'll be adding drop by drop. So what is like in here, this KCl will go inside the cell. It acts like the osmosis process. So what happens when we put comes under like occupational genetics, just to know that. See, you could now I hope like you can understand this thing. Yeah, earlier you might have you come across the 7q21. What is mean by 7q21? Now you can know 7 means the chromosome number. Q means it is the region. This is P and it is Q. This is the 7th chromosome and the Q region. This is so called as band. 7q21 is the band. Okay, 21 is the region, and 21.1 is called as band. This is the enlargement of this position. Okay, and you enlarge this this one, it is called as subband. 7q21.11. That means in 7th chromosome, in the Q position, in the 21 21st region. This is all you have to understand. Earlier, you might have seen these kind of acronym and you might be confused. I hope you are very you can understand this image by this. This is the enlarged I follow this Jinsa trips in band. And even in the light regime, the same thing happened. This is like 18th, just to understand. This translocation you see in leukemia. If you see you have deletion between the chromosome in the Q between 11 Q13, you have a deletion. This is the karyotype formula. Just written. Like getting one will be normal, one will be affected. This designed by the square. One will be normal, one will be affected. Two, anything can come out. If then the generation won't be any problem. This all completely falls on this genetics. This has been carried on from generation to generation. That's what generally before planning for pregnancy, we in terms of abroad countries, we'll be visiting a genetist. They'll be taking these kind of genetic analysis. So whether they have or dominant in or they have a very good genetic disorders, or we can able to identify is there any chance of having a baby with genetic disorders. So accordingly, we can go for the genetic testing. So this is how we do. Whether your father is having like this, whether your father-in-law having any problems, or the grandfather has any problem, they'll be asking all these questions just to know whether they comes under recessive inheritance or dominant inheritance, or the family is very clear, like a normal inheritance pattern. When we have affected father and we have a normal mother, there is a chance of two normal and carrier. So this is the dominant inheritance. The problem of cytogenetic in a place major role. So see, these are all the basics. There are to learn many things to come up. If I want to speak about this for more time, due to a very short time, faster. Sorry. Okay. Why cytogenetics? Why you have to depend on the cytogenetics? Why cytogenetics are important? See, this is cytogenetics helps in adding finding the gene. It all the finding the gene comes under the molecular biology part, and the pathobiology, it all comes under the biologic techniques. So whatever it is, the basic comes from the cytogenetics. We identify the locus. With the help of the locus, we can able to identify the gene, and with the help of the gene mutation, we can understand the pathobiology. What happens to this gene when this has been mutated? What kind of pathobiology? And then we can till hybridization, we have to follow the same technique, and then we'll be adding the probe, and these are all the techniques that we have to follow the probe, because we are running out of time, I think. So I'm sorry. So almost I've taken more than 1 hour, so I don't want to take more time. And this is like a spectral karyotype This is just for understanding, and we can paint the and also for like we can avoid this thing, but just for I'm recent advancement, I think, like last 5 years, like this has been a very like it's like we have a very good results, and very cryptic deletions also you can able to find this. Just you understand like comparative genomic hybridization. This is the same technique which tell the metaphase like and Google that instead of doing some kind of this, like you can take a note on that. Some can take a snapshot, and you can pass to the students, so you get a fellowship with scholarship. So that's the purpose. So you don't want to waste the summer train, like you don't want to waste the summer sitting in the home movies. No problem. Even it should be, it can be what is awesome. Very nice question. Is genetic disorder and chromosomal disabilities genetic disorder and chromosomal abnormalities is same. If you have a chromosomal abnormalities only, you'll be having a genetic disorder. So that is the difference between chromosomal abnormality and genetic disorder. You should have a chromosomal abnormalities like in the patients, and then only like you can able to distinguish the genetic disorder. So if you have chromosomal abnormality, there will be a genetic disorder. So autism, it's like autism. It's a best example is like to understand autism. I can say it by movie. If you see the movie, see the recent Surya movie like 2-3 years back, Pasanga 2, you can understand exact knowledge of autism. You can't say that this is a new disorder. So cerebral disorder. Integrative disorder, and we usually we can able to identify using a questionnaire the clinical features they comes under which kind of autism spectrum disorders. So autism is a kind of a big thing, but autism is a kind of in our view it is frog. In autism view it is horse. So they have eye talent. They can see horse and frog. So that's what we say. Generally, when a child is like when a child is asking my speech.
Really very when you explain about nomenclature.
Thank you.
Thank you
Thanks
so
for the
much.
opportunity.
Now yes, thank you, sir. Now I request Dr. S. Abhinaya, Assistant Professor of Biotechnology Department, to propose the vote of thanks.
A very good afternoon to everyone. It's my privilege to have been asked to propose the vote of thanks on this occasion. I, on behalf of the CAUVERY College for Women, the entire crew of today's international webinar, and on my own behalf, extend a very hearty thanks to all. A big thank you to Dr. Arun Mayaragan for his efforts in delivering the contents of the topic on cytogenetics. I must mention our deep sense of appreciation for his explanation and very lucid presentation.
Thank you.
Thank you, sir. I, from the bottom of my heart, thank the College Management and our Madam Principal for constantly motivating and inspiring us to conduct various activities for the enlightenment of students and faculty. Thank you, ma'am. I extend my heartfelt gratitude to Dr. R. Rameshwari, Head Department of Biotechnology, for being instrumental and supporting us to make this event happen. Thank you, ma'am. I return my sincere gratitude to Dr. G. Gomati, Assistant Professor and Bolivar Club in charge, for coordinating this intellectual webinar. I thank each and every staff member of the Department of Biotechnology for playing key roles in making this webinar successful. Last but not the least, I'm thankful to all the participants from Kaveri College, from outside colleges, from around India, and also from abroad. And thank you so much. I take this opportunity to take the entire crew and the people who have directly or indirectly helped make this event a very grand success. Thank you, one and all.
Thank you, ma'am. And you will receive your certificates within a week's time. Thank you.
Arun sir, thank you very much. Arun sir.
Thank you, ma'am. Thanks for the
Thank you,
opportunity,
sir.
and I would like to thank the participants, and I would like to thank the management and principal madam once again